folks, Dr. Rob Barrington here with some more nutrition advice. I was asked in the week by a colleague what was the connection between raised levels of blood glucose and changes to plasma lipoprotein levels. Now this got me thinking that it would be useful, I think, for those people that are interested in health to understand the biochemical link between these two apparently different metabolic problems. They are very closely connected and they have a common cause. And the common cause is something I've talked about a lot before, and that is the metabolic syndrome. Now, the metabolic syndrome is widely talked about. It is a group of disorders centering on the development of something called insulin resistance. Now, to be able to understand how raised levels of blood glucose and changes to plasma lipoproteins are connected, I'm going to talk through from a nutritional aspect the cause of these two disorders and how they might develop. Changes to plasma glucose levels are quite well understood. The cause of poor blood sugar control is really the consumption of a poor quality diet. There are a number of factors, dietary factors, that can be consumed that will cause a deterioration in the sensitivity of your insulin system. Now, when I talk about insulin sensitivity and insulin resistance, there are insulin receptors on all cells, and those receptors are sensitive to the hormone insulin. When you consume particular carbohydrates, your body releases insulin, and that insulin is there to open glucose transporters on the cells to allow the glucose to go into the cell, and that effectively lowers blood glucose levels. If those receptors become insensitive to the hormone insulin, those transporters aren't opened, they're not present on the plasma membrane, and therefore the glucose is not able to pass into the cell. The glucose stays in the blood, and this causes an elevation in fasting blood glucose levels. And this is the problem that with diabetes. Diabetes and glucose intolerance are conditions whereby you have elevated levels of fasting blood glucose. So the glucose is in your blood, it's not in the cells where it needs to be, and therefore you have some problems because you can't oxidize the glucose if it's not in the cells, and you can't use it for energy, so your energy levels go down, and the glucose also causes damage to the tissues when it's in the blood. The cause of there are a number of causes of insulin resistance that are known. The most common dietary cause of insulin resistance is back to our old friend fructose. Fructose is a monosaccharide that is found in sugar. It's also found in high fructose corn syrup, and it can be found on its own as a sweetener. It's a very sweet molecule, and that's why food manufacturers like it. They can put it in their foods and make their foods very sweet. Many people overconsume sugar. Many people overconsume foods that contain sugar. Now, when you consume fructose, let's say when you overconsume fructose, fructose can only be handled by the liver. The liver is the only organ that can actually deal with fructose. So you're asking a lot of the liver. If you overconsume fructose, you're putting a stress on the liver. Now, there's two things that the liver can do with fructose. It can synthesize glycogen. It can convert the fructose into glucose and then synthesize glycogen, or it can convert the fructose into fatty acids and then into triglycerides. When you overconsume refined fructose, that's refined crystalline fructose that's not naturally found in foods along with fiber. When you overconsume those refined variants of fructose, which would include fruit juices, would include soft drinks, as well as anything with lots of sugar in them, like biscuits and cakes. When you overconsume that type of fructose, the overwhelming amount of energy that you supply to the liver means that it is very unlikely that that fructose will end up as glycogen. Much more likely that fructose will stimulate a pathway called the de novo lipogenesis pathway, and the fructose will flux down that pathway and form fatty acids. Now, these fatty acids are problematic not because fatty acids in themselves are inherently bad, it's because there will be too many of them. If you overconsume sugar or overconsume fructose, de novo lipogenesis pathway is stimulated and there is then a large production of fatty acids. Now, the body will export these fatty acids as triglycerides into the blood. And this is where the link starts between lipoprotein changes and blood sugar. But I'm going to come back to this later. I want to follow through the fructose pathway to where it causes insulin resistance and raised levels of blood glucose. But this is the delineating point between changes to blood glucose and changes to plasma lipoproteins. So we have these triglycerides, they're exported to tissues, and there's lots of them because we're over consuming fructose. We have a poor quality diet. That diet is probably also low in fibre, which means that the fructose and all of the refined sugars that we're consuming from this low quality diet will get into the system super quick, and because they get into the system super quick, that causes extra stress on the liver, which means that the fructose is much more likely to pass down the de novo lipogenesis pathway. The triglycerides will accumulate in tissues as lipids, and these lipids are going to accumulate in skeletal muscle, for example. Now, there is a theory that the accumulation of these lipids in the skeletal muscle actually interferes in some way with the insulin signal cascade. It's not entirely sure how this happens, but that is one of the theories as to how these lipids cause insulin resistance. These lipids accumulate in the tissue and somehow they interfere with the insulin signal cascade, which is the signal sent from the insulin receptor to the inside of the cell to say that insulin has bound to the receptor. So the insulin will bind to the receptor, but the signal from that will not get to the cell, so the cell will therefore think that there is no insulin, and therefore it won't upregulate the amount of glucose transporters, and therefore the sensitivity of that cell to the glucose in the blood has decreased. There is another theory that suggests that these triglycerides are oxidized in the cells. This increases the activity of the electron transport chain, which is the ultimate energy production mechanism in the cell. And one of the byproducts of the electron transport chain is the generation of free radicals. And if you overstimulate the electron transport chain because you have an oversupply of these fatty acids, these resultant free radicals may actually damage the insulin receptors or in some way interfere with the insulin signal cascade. It's not exactly clear what the mechanism is, but there is some way that these fatty acids, when they accumulate in peripheral tissues such as skeletal muscle, they decrease the insulin sensitivity of that muscle, of those muscle cells, and therefore the insulin becomes less effective and less of the glucose from the blood gets into the cells for the production of energy, and therefore your blood glucose levels goes up. Studies have been done on rats where they fed them high amounts of fructose, and they develop, and dogs as well, there's been a number of animal models, but many of the studies have been done on rats, and they develop insulin resistance when you feed them high amounts of fructose within about 2 weeks, which is not a lot of time. Now, some of those studies were initially criticised because other scientists felt that the amount of fructose that was fed to the rats was unrealistic of what humans would have in their diet. Subsequently, a lot of those complaints have died down because more recent studies that have actually evaluated how much fructose some people are consuming have shown that those studies are actually quite realistic for the highest intakes of fructose in certain individuals. So those people that drink a lot of soft drinks, they drink a lot of fruit juice, they consume a lot of sugary products. Some of those rat studies were actually quite realistic in terms of the highest intake of fructose in humans. So insulin resistance is going to develop very quickly. That means that blood sugar levels are going to start to rise quickly. You go through as this metabolic deterioration and insulin insensitivity increases, you may first develop glucose intolerance, whereby you have raised levels of blood glucose, but they're not problematic. And as that develops further, you will actually eventually fall into type 2 diabetes, and that takes decades to develop. But once it develops, it's quite difficult to reverse it unless you are very, very determined to get your health back. So prevention is much better than cure in terms of blood glucose problems. Now, the other problem with a poor quality diet is that not only will you supply fructose, which will drive the insulin resistance, you will also consume lots of refined carbohydrates. You will have a low fiber intake, and that will push up the glycemic index of all of your foods. And all of that all of that starch and all of those carbohydrates that you're consuming will get into the system much more quickly, and that will push up your blood glucose levels. But obviously, if you're insulin insensitive because you're also consuming a lot of fructose, you are going to find that your blood glucose levels are going to go up very rapidly postprandially, and they're not going to go down because you don't have any sensitivity in your insulin receptors, and therefore your blood glucose levels will stay high. So it's the combination of fructose with other refined carbohydrates that are really problematic at causing blood glucose problems, and that's a bit of an overview. Now, another thing that is problematic for these triglycerides is that they will also accumulate in the liver. Not only are they exported to peripheral tissues like the skeletal muscle, they will also accumulate in the liver, and that causes the formation of something called non-alcoholic fatty liver disease. Now, non-alcoholic fatty liver disease is pathologically exactly the same as alcoholic fatty liver disease, and that's because of the similarity between the fructose molecule and the ethanol molecule and how it's metabolized in the liver. They generally both result in the formation of fatty acids, and those fatty acids ultimately accumulate in tissues, and in particular they will accumulate in the liver. Now, this is currently being researched. It's only recently been understood how many people actually have non-alcoholic fatty liver disease and how common it is. One of the symptoms of having too many of these fatty acids and this accumulation is an increase in waist circumference. You tend to get an accumulation of fat around the waist. But those people with, who are generally not overweight but have this accumulation of fat around the waist generally are going to have this non-alcoholic fatty liver disease. That is an outward symptom of this inward unseen disease developing. Now, as these fatty acids develop, as these triglycerides accumulate in the liver, they can develop into something called steatohepatitis, which is a deterioration of the liver's function, and ultimately you can get cirrhosis of the liver just as you would if you were consuming alcohol. Now, one thing to say that obviously if you've got this accumulation of fat in the liver, you're going to affect the metabolic regulation of the liver. Many animal studies have artificially induced this a non-alcoholic fatty liver and then measured changes to certain metabolic pathways, and it's clear from animal studies that there is a deterioration in amino acid metabolism, there is a deterioration in carbohydrate metabolism, and there is a deterioration in fatty acid and lipid metabolism when these fatty acids accumulate in the liver. So here we have the central problem in metabolic syndrome. It's a deteriorating liver, and your liver is your metabolic factory that processes most of your nutrients and food. So when you have these problems, this development of non-alcoholic fatty liver, which one of the symptoms would obviously be raised levels of blood glucose, you're also going to have changes to all of the metabolic pathways that the liver is involved in. And many studies have looked, for example, at changes to the amino acid levels in plasma, changes to the fatty acid levels, and surprisingly, changes to lipoprotein levels. And this is where the link to lipoprotein levels is really found. And it's this underlying metabolic syndrome, this insulin resistant liver damage state that is really the cause of both of these conditions, and it increases the risk of diseases such as cardiovascular disease, type 2 diabetes, obesity, and this raft of these diseases that are all associated with this underlying metabolic problem. So if you go back to the point where the fructose has passed through the de novo lipogenesis pathway and has produced fatty acids, they are packaged into a type of lipoprotein called a very low density lipoprotein particle. These VLDLs are actually measured in the plasma as triglycerides. So if you've had your triglyceride levels measured, you've actually had your VLDL levels measured. They're synonymous, they're the same. These very low density lipoprotein particles export mainly triglycerides, which is why they're called colloquial triglycerides. They export triglycerides to the tissues. If you are consuming a lot of fructose and you've got a stimulated de novo lipogenesis pathway, obviously you're going to be producing a lot of fatty acids. They're going to produce a lot of triglycerides, because a triglyceride is 3 fatty acids joined to a glycerol molecule. They are packaged into these VLDL particles and then sent to the tissues, and that's how the triglycerides get to the tissues. Now, as these VLDL particles are unloading their triglycerides in the tissues, their density changes, and they will pass firstly into, they will be classified as an intermediate density lipoprotein, and then as the density falls further, they will become a low density lipoprotein particle. And this is the connection between why you get increased levels of LDL particles when you tend to have metabolic problems with the liver, because LDL particles are really just VLDL particles that have had their triglyceride levels removed. So you would expect somebody who had elevated levels of triglycerides also to have elevated levels of LDL. It's very often what is found. There is a classic profile that occurs in lipoprotein changes, and generally those people who have high levels of LDL will also have elevated levels of triglycerides as well. Now, it's worth mentioning here, and it's a little bit of a kind of a tangent, but the classic way of measuring cholesterol is really not very helpful. Total cholesterol is a measurement of all of the cholesterol you have in your plasma, and it's really not very helpful, because some of the cholesterol that you have in your plasma is actually going from the tissues back to the liver to be excreted, and this is where the high density lipoprotein particle comes in. The high density lipoprotein particle is a particle synthesized in the liver. It goes into the peripheral tissues and it collects various various chemicals off other lipoproteins, including cholesterol and some fatty acids, some lipids as well, and it transports them back to the liver for excretion. So elevations of high density lipoprotein are actually beneficial. So when you measure total cholesterol, it's not really that very helpful, because it includes all of the cholesterol carrying particles, and therefore it doesn't really tell you what your status is. For example, if you have high levels of HDL, high density lipoprotein particle, that can actually be protective of cardiovascular disease, because although the cholesterol is in the blood, it's actually being transported back to the liver for excretion, and therefore it's not going to cause any problematic metabolic changes. So total cholesterol is not really very useful. Then we go to the next level up, where we start to differentiate between the density of the particles, and this is usually done with ultracentrifugation, and the particles have different densities, and that's why they've got different names. The reality of this is that the particles are not actually different. It's a grey scale all the way from the densest to the most dense to the least dense. So although they've got different names, they're actually ranges within an ultracentrifuge column, and therefore it's actually quite difficult to name some of these particles sometimes. But when you look at the ratio of the LDL to the HDL, that's more informative. If you have high levels of LDL and low levels of HDL, that is a higher risk. That means that you're a high risk of cardiovascular disease, because the LDL is transporting the cholesterol to the tissues, where you don't really want an accumulation of too much cholesterol, and the HDL is transporting it away from the tissues. So if you've got more cholesterol going to the tissues and less cholesterol coming away, that indicates an accumulation of both triglycerides and cholesterol within the tissues, and that is indicative of a metabolic problem. So the ratio between the LDL and the HDL is more useful But there are problems with that as well, because the LDL particle is not a single particle and the HDL particle is not a single particle. The LDL particle, for example, can be split into the large buoyant type of LDL and the small dense type of LDL. Now generally, if you have raised levels of the large buoyant type of LDL, you don't have an increased risk of cardiovascular disease. You have to have the increases in the small dense LDL particle to have an increase in the risk of cardiovascular disease. And there are also different densities of the high density lipoprotein particle as well. So your basic biochemical ranges that you get from a typical clinical cholesterol test are really not helpful at all, because it's based on scientific and it's based on a scientific understanding of lipoproteins from 40-50 years ago. Lipoprotein chemistry has moved on massively in the last 20 years and even further and more rapidly in the last 10 years, and the cholesterol tests that you get done at a doctor's are not really representative of that modern understanding of lipoproteins, and therefore I would suggest that most cholesterol testing is actually quite pointless. Certainly total cholesterol is completely meaningless. It doesn't have any relation to anything. When you start to look into it, some people are actually protected from having high levels of cholesterol in their plasma. It depends on your age, it depends on your sex, it depends on what other things you're doing. So cholesterol testing, even the ratio of LDL to HDL, is quite misleading unless you take into account these other particles. And there are actually subgroups of low density lipoprotein particle. For example, there's a lipoprotein A particle, which is a type of, I think it's a genetic variant of the low density lipoprotein particle, and that's a very atherogenic particle, and it has a very high levels of lipoprotein A are very indicative of a high risk of cardiovascular disease. So, and that's often not even measured. It was Linus Pauling that showed that levels of lipoprotein A are actually inversely associated with your plasma vitamin C levels. So this is far more complex than is made out by doctors. Whether doctors, I'm sure some doctors do understand this, but I'm sure the vast majority of them have no idea that there's this complexity. They're just given a test, they're given the results, and they're told how to interpret that test. Unless you really go into the chemistry of lipoproteins and understand them, and even the research scientists that are researching this don't fully understand how these particles are related to cardiovascular disease. So you have to be very careful how you interpret these levels. That being said, the metabolic syndrome does cause changes to these levels, and those changes, I would suggest, are detrimental. They're certainly indicative of detrimental changes if you look in the literature. Now, another thing that will occur, now obviously you've got an increase in very low density lipoprotein particles and low density lipoprotein particles if you have an increase in flux through the de novo lipogenesis pathway. Another reason that you may get alterations in cholesterol metabolism when you have metabolic syndrome is this general accumulation of fatty acids in the liver that causes a general deterioration of metabolic pathways. Remember, the liver is the processing factory of all of your really all of your nutrients in your blood, and it's one of the processing and detoxifying organ that will help excrete cholesterol. There are many receptors for various types of cholesterol and various types of lipoprotein, and obviously changes to the metabolism of the liver can cause metabolic problems. Now, let's talk about the high density lipoprotein particle. When you tend to get the metabolic syndrome and your blood glucose levels go up, and your very low density lipoprotein particles, that's your triglyceride levels, go up, and your low density lipoprotein particles go up, we've explained all that, why that might occur. What tends to happen is your high density lipoprotein particle level concentrations in the blood will go down. Now remember, this is the particle that will take the cholesterol and other lipids and take them to the liver for excretion. If levels of HDL go down, that is generally seen as a bad metabolic change. Now, when you get this underlying metabolic problem with the liver, what also develops is an inflammatory state. Non-alcoholic fatty liver disease is associated with inflammation, and you can measure that inflammation by measuring systemic markers of inflammation such as C-reactive protein. So those people with metabolic syndrome tend to have high levels of this C-reactive protein, showing that they have this inflammatory condition. Now, inflammation leads to oxidative stress. It leads to the generation of free radicals. Now, one of the functions of the high density lipoprotein particle that is virtually never discussed and is never mentioned is that it is able to take from the low density lipoprotein particles, it is able to take away lipid peroxides. Now, lipid peroxides are fats that have been altered by oxygen so that they're now in a problematic state in terms of the fact that they could cause the generation of free radicals if they're incorporated into tissues and into cell membranes. One of the functions of the high density lipoprotein is as a type of antioxidant. It takes away these damaged and altered lipids from the low density lipoprotein particles before they're incorporated into tissues, and in that way it prevents them being placed in tissues where they could generate, where they would either not function correctly or they could generate further free radical reactions. So the HDL particle actually acts as a type of antioxidant, and those lipid peroxides are then taken to the liver where they're metabolized and excreted. If you have very high levels of LDL and you also have a very poor diet and you also have non-alcoholic fatty liver disease, so you have general oxidative conditions for general oxidative stress in your body, you will have lots of lipid peroxides in your low density lipoprotein particles. I would suggest that this overloads the high density lipoprotein particles with oxidative stress, because they will accumulate these lipid peroxides, which are in themselves generating, they are free radicals themselves, they will generate free radicals further. If you have a poor diet, your antioxidant status is low, you've got inflammation, you've got too many low density lipoprotein particles, you've got too many lipid peroxides in those low density lipoprotein particles, the HDL particles will become overwhelmed and therefore they will start to suffer from oxidative stress. Now, there is some evidence, if you go to the literature, that this general oxidative stress and inflammatory state is detrimental to the function and the structure of these high density lipoprotein particles, and this may be the reason that HDL decreases when you have this underlying metabolic syndrome. So therefore, what you would expect is with metabolic syndrome, your blood glucose levels will go up, your very low density lipoprotein levels will go up, that's your triglyceride levels, your low density lipoprotein levels will also go up because they're derived from the very low density lipoprotein particles, and the general oxidative stress will cause a decrease in your high density lipoprotein particles. And that's pretty much the profile you see with the development of the metabolic syndrome and this non-alcoholic fatty liver. Now, to test, if you go to the literature and you want to, you'd want some evidence for the fact that it's oxidative stress that causes a decrease in the high density lipoprotein particles, you would expect the taking of antioxidants such as vitamin C to increase levels of high density lipoprotein particles, and that's exactly what you find. Many of the nutrients that you take that are antioxidants cause an increase in the high density lipoprotein particles, and it's probably that when you take those antioxidants, they're providing a way for the oxidative stress to be reduced slightly, and that takes the stress and reduces these lipid peroxides, and therefore there's less damage and less reduction in the high density lipoprotein concentrations in the plasma. So that's a general overview of where blood glucose levels link to lipoprotein metabolism. It's very complex and it's multifactorial, but there is a clear link there, and that clear link is the metabolic syndrome and the development of non-alcoholic fatty liver, and that comes from poor diet. Now, changing your diet is the one thing that you could do to avoid all this. The cause of all of this problem, the cause of all of these metabolic changes, is fructose. It's fructose, which is found in sugar, which is sucrose. It's found in high fructose corn syrup, and it's also added to, as sugar, it's added to cakes and biscuits and all the things that people know they shouldn't eat. One thing people often ask is, why is fructose in fruit not detrimental? Well, fruit contains high levels of fibre, and that fibre is protective of metabolic disorders, because the fibre decreases the rate at which the fructose can get into the liver. If you can reduce the speed that the fructose goes into the liver by consuming high amounts of fibre, what you'll effectively do is you will slow the rate of de novo lipogenesis, and therefore more of the fructose is likely to go into the production of glycogen. And this is one of the reasons why exercise is also protective of the metabolic syndrome, because you deplete your glycogen, that means the fructose that you do consume is much more likely to go down the glycogen pathway than the de novo lipogenesis pathway. So most of the strategies nutritionally that decrease the risk of metabolic syndrome are actually decreasing the flux through the de novo lipogenesis pathway. And there's many different nutritional strategies that you can employ to be able to prevent that. And they all seem to have this common ability. So, for example, increasing the consumption of oats. Oats have high amounts of fiber. Whatever you consume with the oats, even if it's honey or it's sugar sprinkled on the top, that fiber is going to sit in your stomach for a while. It's going to sit in your small intestine. The fibre is going to inhibit the sugars getting to the unstirred layer of the enterocytes, and that means that they're only going to trickle into the blood. And as they trickle into the blood, if you've also been performing exercise as well, when they get to the liver, they will start to resynthesize the glycogen that you've used during your exercise, and then the liver is much more able to cope with the levels of nutrients being sent. It's being sent from the hepatic portal vein, and therefore it's much more less likely to stimulate and overstimulate the de novo lipogenesis pathway. And this is really the crux of understanding how to look after your health in terms of preventing metabolic syndrome and cardiovascular disease, and these associated disorders such as obesity and type 2 diabetes. So high fiber diets are protective. Food in its raw unrefined state is protective. As soon as you start refining out the fiber, as soon as you start creating these very purified sugars, as soon as you start consuming lots of refined carbohydrates, you start to stimulate this de novo lipogenesis pathway, and this is the root cause of all of these problems, and this is how high blood glucose levels relates to changes in lipoprotein metabolism. So I hope that was interesting. It's an overview. It was very brief. There's much more to this. I do suggest you go to my blog. There is a primer on lipoprotein metabolism, which is very useful. If you search in the box for primer lipoprotein metabolism, you'll find it. I will also put the link in the description box for this video so you can go to it. It's an overview of how the lipoprotein metabolism works, and I think that's a very good thing to have as a background information, so that when you read other articles on the internet, you have an understanding of what these lipoprotein particles do and where they go and what they're converted into, and that's going to help you understand the best nutritional strategies to look after your health. So I hope that was interesting. Please subscribe to my channel if you haven't done so already, and please like this video if you thought it was helpful. I will see you soon for another video, and in the meantime, don't forget to please eat well, stay healthy and protect yourself, and I'll see you soon.